In this final section, we're taking a look at the ventricles of the brain to begin with. So we notice this view here of the brain, and in the blue shading, it's showing what represents hollow cavities within the brain that continue down the spinal cord. Or here, you notice with this frontal section, we can see again these hollow cavities known as the ventricles, of which there are four major ventricles of the brain. And the cavities themselves are now in turn filled with cerebrospinal fluid. So this fluid's in circulation within these cavities of the brain. And the source of producing cerebrospinal fluid we mentioned earlier, that key neuroglial cell, an ependymal cell. So these are present bordering each of the four ventricles, and of course playing a major role in producing that cerebrospinal fluid. So the exact location where CSF is produced is known as a choroid plexus. And a choroid plexus has two major parts, of course, the ependymal neuroglial cell. Its function is to filter plasma of our blood, and the source of that blood plasma are capillaries. So the union of capillaries and the ependymal cells together, that is a choroid plexus. And that's the exact source of producing cerebrospinal fluid. And truly due to that filtering process that's done by the ependyma, this is what makes the plasma of our blood quite unique from cerebrospinal fluid from which it's produced. And again, each of those four major ventricles has its own designated choroid plexus. So here's just a simple diagram to show that there is this distinct route of circulation. We're not concerned with learning the pathway, but you notice each of those ventricles has its own dedicated choroid plexus. And cerebrospinal fluid will flow through those ventricles in and around the brain and spinal cord. And a closer look, here we see a capillary, the source of blood plasma, and represented by these pink cells lining the capillary. These are the ependymal cells. And having the union of these two together, we're looking at a choroid plexus. So ultimately, the filtered plasma flows out, and now we've produced cerebrospinal fluid. Another topic here at the end of this unit, we've already covered in the muscular system, and so we're now relating it back to the nervous system. We had this term earlier, a motor unit. So a single, at this point we could say somatic motor neuron that's now signaling to a distinct number of skeletal muscle fibers. And taking a closer look at that interaction between the neuron and the muscle tissue, right here at this region known as the neuromuscular junction, we can see, of course, now the terminal boutons, the synaptic terminals of a somatic motor neuron. This is the location where now the vesicles containing neurotransmitter acetylcholine, ACH, are being released from the synaptic terminals. The chemical diffuses that distance between the synaptic terminals and the sarcolemma. And now it's on the sarcolemma of the muscle cell. There's receptors for the neurotransmitter. And ultimately, that signal is what stimulates the muscle to begin its phase of contraction. So we're looking right at that location where the nervous system is now directly communicating to the sarcolemma of the skeletal muscle fibers. And we saw that with one of our tissue samples. So we can see those telodendria leading down to individual terminal bouton. And here we see, of course, a signaling to this particular skeletal muscle fiber.